Okay, we are going to work on database mining today. So what kind of database mining that we are going to do? So it is really important that you know how to mine or get information once you get your sequence. For example, right, Assalamu alaikum. Okay, so for example, you go to NCBI, this website, and it's National Center for Biotechnology Information. So this website allows you to submit, if let's say you have a sequence, you can submit your sequence and it becomes a small publication. And if let's say you need to have a sequence, so as a reference to do, let's say you want to do a primer, so you can download your sequence from NCBI and you can learn, develop, analyze, and do research. If let's say you are looking for an unknown gene, so what you can do is you can take from several types of, if let's say the gene, in my case, I was looking for a streptozidin synthase in Kerato, which has never been found at that time. So what needs to be done is you need to find lots of genes that are close to Metrogyna and align them and see where do they meet, and then from the meeting point, from where they are aligned, you should be able to find a specific place with a conserved region. So what is conserved region? Okay, I'm gonna show you afterwards. Okay, let's say this is and then you have blast. Let's say there is blast. If you go to NCBI, okay, this is what we call blast. Okay, right here, and you click on blast, and then it's gonna come out like this. Okay, let's say I just got my sequence, and let's say I have sent my sequence, my DNA to, let's say I sent my plasmid to First Base, which is a company in Serdang, and then so they helped me sequence my DNA, and they come up with a result coming from that sequencer. Okay, so they emailed me the results, and what I need to do is I downloaded them, and so basically this is what I got from what they the sequence they gave me is from C up to A, which is if you see how long it is, it's about you can take a look at your PowerPoint. It says how long it is. It's about 1257 base pair. Okay, so you can just copy this, and the next thing you need to do is go to your NCBI BLAST. You can go to, you can see there are two types of BLAST right here. BLAST X, translated nucleotide into protein, and T-BLAST10, protein translated nucleotide, and then you have, oh sorry, I said two, there are actually four. You have Nucleotide Blast, which is from nucleotide to nucleotide, BLAST X, translated nucleotide to protein, T-BLAST10, protein to translated nucleotide, and Protein Blast, which is from protein to protein. So, because my gene was a nucleotide, so we go to Nucleotide Blast. Okay, it's gonna be a while. Right. Okay, so we have gone into the website, which is Nucleotide Blast. We have like several choice that you can make. Let's say you have a sequence from a DNA sequence, you can just click on blast. If you have a protein sequence, let's say you have sequenced your protein, you can go to protein blast. But if, let's say, you have a nucleotide sequence that you want it to be translated into protein, you can do blast X. And if it's a protein that you want it to be reversed back to translated nucleotide, you can use T+10. Okay, so it's gonna come out like this. This is the website. So it says here, enter accession number or FASTA sequence. Okay, so I have copied my sequence. You can just pasted it in right here, and then you can just everything is just normal. You want to find it. So you want to look for something that is highly similar. And so you can choose whichever you like, or somewhat similar. So you can just press plus. Okay, so after you press plus, it's gonna take a while because it's doing it's searching for your database and trying to match everything up and see which one it matches. Okay, so I'm gonna wait for a while and let you digest the information. Alright, so it came out as something like this. So you have Mitragyna species, strictocytin synthase, mRNA, complete CDS, which is okay. So you have the score, the total score, query cover 100%. So you have percentage of identity, which is 100%. That means it is definitely my Mitragyna species. So the ones that I have just keyed in was definitely Mitragyna species. And it has also some 98% of similarity with other types of Mitragyna, which is about the same. If we go in reverse, if let's say you choose somewhat similar sequence, okay, and you press blast, let's see. Okay, it takes a while. Okay, in the meantime, sorry. Okay, there is some error. So okay, if let's say you are using some other, because that was really similar, so probably they couldn't find others that is a little different. So let's move on. So if let's say you have other that you want to find, like this one, let's say one. Okay, so we had it about 100%. So that means it's really it's identical. It's the same thing. That means it is that thing. Okay. So if let's say you have something that has less similar, okay. Which is about, let's say, it's 70% or something. So that actually tells you that it is probably similar to something, okay, but it's not identical, okay. And these are actually called the sequence homology, okay. And okay, so and like I said before this, if let's say you have no idea on what is the gene, what is the sequence of the gene, and you want to start looking for that particular gene, you can go to NCBI, let's say, and click on nucleotide. Why are we looking for nucleotide? Is because it helps you to find a sequence similarity with homology. So let's say you want to use MAP kinase. Okay, we're going to search for MAP kinase. Okay, so there are tons of results for MAP kinase, and you can take a look at results by taxon. So the tree can choose to which one that is closer to your material. For example, if your stuff is a plant, so these shouldn't be something that you should click on if, let's say, you want to know an exact the sequence. Okay, so you have FASTA. You should go to the FASTA, and then okay, and then what you need to do is copy from this side to the end. Okay, and you can get all those sequence. And like I said, if let's say you're looking for sequence alignment, ClustalW. Sorry, it's ClustalW. So this is for multiple sequence alignment that you can help. You can, let's say you put 5 in here, and you just copy it from this one. Sorry, this one. Okay, you just copy it. Let's say you take 5, and there are. Let's say you take 5 of these. Okay, this is too long. I'm gonna take smaller ones. Wow. Okay. Wow, that's too big. Probably need to find from some bacteria. E. coli, maybe. I'll choose which one. So you can find. You can. Let's say you put all of the sequence. Let's say you put one something and blah blah, and then next one and something like that. And okay. Let's say we do the. Let's say we put A to C to A, C to random, and GTC. Okay, that's the third one. Okay, so let's say this one you put 1. Then we put 2. Okay, and then we put 3. And we put 4. This is just an example, just to show you. Okay. Alright, so execute multiple alignment. Let's see how this is going to come out. And alright, so the result is going to show you that this is how it looks like. There is a star right here. That means everything is conserved. When you have a star, that means all of them are the same, which is A. And star, there is a C right here. And if you have dots, like 2 dots, that means it's quite similar. And we have 1 dot, 1 is different. Something. Okay. So that's how you use sequence alignment, which may help you to construct a primer if you want to start it to begin with 0 information on the sequence. Okay, but actually it is quite hard if, let's say, you are working with new organism, for example, lemongrass, or probably the ones that originates from Malaysia. So probably it's going to take a while for you to find it in the database, or probably it's not even there. So you need to find ways to figure out on that particular gene. So there are many methods actually that you can use, but that is one of them that you can use. Okay, it's not easy, but it's something that you can use. All right.